All right, so the next is flagella. And we talked about this in class that it is for movement of the bacteria. And for bacteria, there are 4 flagellar arrangements, or maybe 5, but 4 like outer arrangements. Flagellar arrangements. Arrangements. Okay. And I'll just draw them out, and then we'll talk about how you just have to know how 2 of them move. So the first one is called monotrichous. And on the exam, you can just like name it and just put like the picture if you want. So it's 1 flagella from one end of the cell. The number 2, or the second one, would be lophotrichous. Okay, and then that is many flagella from one end of the cell still. And then number 3 is amphitrichous. Okay, and that's going to be 1 flagella from each end of the cell. All right, and then the last one here is the peritrichous. Okay, that's going to be many flagella kind of all over, dispersed throughout the cell, around the cell. Okay, so the movement can change depending on the type of arrangement. So the ones that I want you to know for movement is going to be peritrichous and monotrichous. So I'm going to put that, or I'm going to show you the slide that has these 2 movements. So make sure you know the movement. Okay. Oh yes, and then I also forgot to mention that gram-negative bacteria is only 2 to 3 layers of the peptidoglycan. And if we measure those layers, it's going to be around 8 nanometers in thickness. And then these like 15 layers is going to be around 20 to 35 nanometers, like if you actually measure it. Okay. They are just like these long structures that come out, and you can see this one's flagella all over. So this is a peritrichous. Here's the tiny little fimbria hairs. Those do not move, so they do not move, these ones, but the flagella move in a very specific way. And let me show you that. So we have these are the different arrangements. And then you can see monotrichous and then peritrichous. Look at these crazy things all over. That's really cool. So then these are the 2 movements you have to know. So polar is another term for monotrichous flagella. So the flagella themselves will rotate counterclockwise or clockwise. So CCW stands for counterclockwise. And then CW is clockwise. So essentially, clockwise is to the right. And then counterclockwise is to the left. Okay, so you can see them turning. So that's going to be the flagella actually rotating. And
the long whip-like portion of the flagella.
Okay, so, I just want to show you how the flagella is rotating. And you can see there's this whole structure in and that's embedded into the membrane. And the flagella itself, though, is like a rotor, and it will rotate in different directions, and that causes the overall cell to move in different directions. So that's what we're talking about when we talk about counterclockwise or clockwise. So if you look at this one, you can see that there is a clockwise rotation. You see that there's a clockwise rotation of the flagella, and that's going to cause all of the flagella to move behind it for peritrichus. And then that's going to make a movement of run. So it's going to go forward. And then if there is a clockwise rotation, so they move to the right, then all the flagella kind of go all over, and it causes the cell to just tumble everywhere. Then they can repeat these two counterclockwise and clockwise rotations to get to the area they need to go. Tumble is kind of like a turn, and then run is just going straight forward. Then this is the monotrichus. It just has one flagella. And when the monotrichus flagella rotates counterclockwise, CCW, to the left, then the cell will go forward. And this one doesn't have any tumble movement, but instead the opposite rotation is just going to make it go backwards, and then it can kind of turn to the one side and then go like basically backwards and forwards and like rotate that way. So there's no like tumbling like in the flagella, like in the peritrichus, because there's only one. And so it either runs forward with the counterclockwise rotation, cell runs, and then cell can reverse for clockwise rotation. So this is for the reversible flagella. And then there is another one down here where the clockwise rotation, the cell basically runs the whole time, but just turns around. Okay, so there's kind of two ways that the flagella can rotate, but I just want you to know like the reversible flagella and its relation to peritrichus. Like for peritrichus, for both of these, the run movement is counterclockwise rotation, and then this one counterclockwise rotation is reversing, so it goes backwards if the flagella goes the other way, and then this one clockwise rotation is a tumble. Okay. All right, so this is E. coli swimming, and I'm just going to show you here. Okay, so as you can see, if I could zoom in, this is E. coli down there running. Let me repeat that. So if you look at the top left, you can see a bunch of tumbling. And then if you look at the E. coli on the right side, right, on the right side, you can see it was tumbling, and then it runs all the way down with all the flagella behind it. It's quite amazing. Okay, all right, the next one is going to be Archaella. So Archaea have a flagella, but it's not called flagella. Archaea have something called Archaella. So this is for movement of the archaea, and essentially in order to uses ATP to move it, and it's like a thinner, not as thick as the bacteria flagella. So for this one though, I forgot to mention for bacteria flagella. So bacteria will use proton motive force to rotate flagella. Okay, so I'm going to show you that in the slides. So we have the flagella here, and you could see that it's rotating. But really what's rotating is the same thing that helps ATP synthase to rotate in the electron transport chain. Is there's an accumulation of hydrogen ions, right, or protons, sometimes called protons, and then these will accumulate outside the cell or in the periplasmic space, and then it will move through this giant protein complex that's attached to the flagella, and like water falling down like the little turbine, right? It'll turn the turbine as these hydrogens flow into the cell, and it's really cool. So this is the only thing that is causing the movement of flagella. No ATP required, and this is an actual picture of this like rotor, hook, rod, and MS ring. Like all these structures are in the bacteria, embedded in the membrane, and then they rotate as hydrogens flow through it. And hydrogens flowing down their concentration gradient, so higher amount of hydrogens naturally want to go back in, so to the less concentrated area, and then this is called, right, this is called chemiosmosis. So the movement of chemicals down the concentration gradient, so going from high to lower concentration, and then that's a proton causing a proton motive force. So the protons are moving and causing the movement again of the flagella. And we talked about this last time, but we talked about H. pylori using its flagellum to invade the stomach membrane. And remember, the flagella allowed it to move, the H. pylori to move really fast towards the epithelium. So the stomach, the lining of the stomach, it moves through the acid so quickly, and then it can create the urease enzyme that we tested for. And then that urease creates a neutral bubble to change the pH of the cell of the stomach, and then that allows it to replicate. But without the flagella, it wouldn't be able to swim fast enough, and it would be burned up by the acid. So a lot of bacteria get past your stomach acid by quickly, like literally swimming through your stomach acid fast enough where the acid doesn't destroy the cell. And then it ends up on your mucus right here, right? This mucus area, and it's kind of more safe now from all of the acid in your stomach. All right, the last flagella that I wanted to mention again about for spirochetes is they don't have an outer flagella. So this is an endoflagella or periplasmic flagella. So this is not a worm, right? This is a bacteria, and it has the flagella inside, but the flagella will still rotate inside. So when the flagella rotates inside, then the whole cell will rotate. And this is the corkscrew motility. And this is how we talked about the very first day of class. This is how the bacteria that causes syphilis, it will rotate its flagella inside and move like a corkscrew and kind of drill its way into your cells. And so endoflagella are the main thing allowing the syphilis to cause infection because of this movement and causing the whole cell, which is already spiral shaped, right? It's already a shape of a corkscrew. And then if you rotate it, then of course it's going to be able to drill into your cells. All right. So the last one on here is the pili. We did say fimbria are the small hairs, right? Small hair-like Velcro structure. They don't move. So if they're called fimbria, they do not move. However, bacteria do have different types of pili. So we have this is the fimbria. These are short pili. But then there are the long pili. So the long pili of bacteria, we talked about one in class called the sex pilis or pili, and those are for transferring DNA, plasmid DNA, in conjugation. So these are used for a process called conjugation. Bacterial conjugation is when it uses the pilus, brings the two bacteria, right, are brought close together by the sex pili, and then it's able to transfer the DNA, plasmid DNA, once they get close. But without the pilus, they wouldn't be able to transfer in this way. Okay, and then there's another one called type 4 pilus, and there's type 3 as well. And these type 4 are going to have a specific movement called, the jerking, like a jerking motion. So they extend and retract, extend the pilus and retract. And it causes them to, the cells to move together, because they're all kind of tethered together with this rope-like structure. And then it allows, when they're all tethered together with this little rope, right? And then this rope will then extend and then retract, and it like moves back and forth. And so then they can all move in one direction together. There it is. All right, so this is the twitching motility of Pseudomonas. So Pseudomonas has a type 4 pilus, and it's going to do twitching. So in green here, you'll see those are the points of release where the pilus is being released. And you can see right here on the bottom right, they're all kind of moving together. You can see some twitching back and forth. Go back one more time, and then there's ones up there on the top left, and you can see them like literally twitching and not moving anywhere. Again, that's because the pilus that's connecting them in between each cell. And then this crazy group of Pseudomonas right here in the middle, they're like all mostly green, and they're moving all together because they're connected with the pilus, and that's crazy. You guys always wonder, right, when I'm like, oh, we're using Pseudomonas, it causes skin infections, because look how well they stick together and they move together. And that, imagine that like being stuck on your skin. If you have a cut, it like really stays really well in the cut. Okay, so this movement overall of Flagella is going to have a net movement towards an attractant and or movement away from something. So when we have chemotaxis, this is chemotaxis is the movement toward or away from something. So chemotaxis is the cell movement. So it's moving its flagella, and then it's going toward or away from a specific chemical. It could be sugar too, or chemical, right, or macromolecule. So if it's positive chemotaxis, that means it's going toward, so it wants that thing like food. And then if it's negative chemotaxis, then that's going to move away, right? So it's going to move its flagella away from that chemical. So we're going to skip that, but I wanted to show you some different chemotaxis. So there are ways that bacteria can respond with their flagella in their differences in the response to oxygen. So they'll move their flagella. For example, E. coli will do more run movements in the presence of oxygen. And then E. coli will do more tumble movements in anaerobic environment, so no oxygen environment. Okay. All right. And then one other one that is just, I wanted to show you the video Pseudomonas here, moving toward a sugar molecule. So you can see here, it's going going to go towards the sugar molecule. Eventually, they kind of go back. This is a directional movement of a monotrichous. Look, they're all congregating, right? So they all are Pseudomonas. And this one right there, that guy, he came back. That one at the very end, he came back as well. So